Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I'm the marketing manager at Medicinal Genomics and proud member of the team that puts on the CannMed conference every year. All right, let's address the elephant in the room. If you've signed up for email alerts, then you should have received an email yesterday with an important update for CannMed 2020. If you have not signed up for email alerts, then please go to cannmedevents.com/coffee talk and do so now. The news is that our team has made the difficult decision to postpone this year's event to the spring of next year. CannMed 2021 will take place April 12th through 14th at the Pasadena Convention Center in Pasadena, California. If you're a faithful listener of the podcast, you have heard me say that we were closely monitoring the COVID-19 situation in California and working with the Pasadena Convention Center and local leaders to make sure we can provide a safe environment for our staff and attendees. Our team felt moving the event to April 2021 would be the best way for us to provide a safe environment for everyone. We are offering a 100% refund to current ticket holders should they have a conflict with the new dates. Go to cannmedevents.com/COVID-19 for more information. The good news is the CannMed Coffee Talk podcast will continue with new episodes every other Wednesday. We look forward to continuing the CannMed conversation through this medium, as well as on social media and on our website. We will be announcing our safety keynote presenter, an expanded Phase 2 schedule, and new speakers, as well as multiple panel discussions throughout the rest of this year and into 2021. So please sign up for email alerts if you haven't already. That link again is cannmedevents.com/COVID-19. And also follow us on Instagram, Facebook, Twitter, and LinkedIn. Just search for CannMed Events. Now, with that out of the way, let me tell you about today's guest, Keith Allen. Keith is the Director of Bioinformatics at Front Range Biosciences, where he is tasked with exploring the cannabis genome to find genetic markers that can be selected in breeding projects. His current focus is on cannabinoid and terpene production, but as we discuss in our conversation, there are a number of different traits cannabis and hemp breeders will want to select for in the future, including yield, drought resistance, pathogen resistance, flowering time, hardiness for the outdoor environment, and more. The opportunity for improving the cannabis and hemp plant is only restricted by our understanding of the genome, which Keith readily admits is still in its infancy. Keith will be presenting on gene families and gene networks underlying oil production in Cannabis Trichomes at CannMed 2021 this spring. So please do consider picking up your ticket for the event now. Before we get to my conversation with Keith, I want to thank this episode's sponsor, the Growers Network. Growers Network is committed to providing the highest quality cannabis content, education, and resources. Be sure to check out their Canna Crib series, your behind-the-scenes look at some of the most sophisticated, technologically advanced grows in the world. Visit them at youtube.com/growersnetwork. And finally, this episode of the CannMed Coffee Talk podcast is fueled by the Hemp and Coffee Exchange. Hemp coffee is a healthy, delicious, natural product rich in trace minerals and nutrients, providing sustained energy without the crash of regular coffee. For more information, check out hempcoffeeexchange.com and use the promo code DRINKHEMP to get 10% off your purchase. Okay, without any further ado, please enjoy my conversation with Keith Allen from Front Range Biosciences. Good afternoon, Keith. Thanks for joining us today.
Good afternoon, no problem.
Excellent. And thank you so much for agreeing to be a part of CannMed 2020. I know the team here at Medicinal Genomics has been an admirer of your work for quite a few years, so we're really excited to have you presenting this year.
Yeah, I'm excited to be a part of it. This has been on my radar for a while. So
Now, have you ever attended CannMed before, or would this be your first?
this will be my first.
Excellent, excellent. And we already have you secured and a speaking slot. I was wondering if you could talk a little bit about what you're intending to present to everyone.
So what I'm going to be talking about started out as being about gene networks underlying oil profile in cannabis trichomes. And it turns out I got a little bit distracted on the way to getting there. And so what I'm going to talk about is really the gene families. So before I could get to networks of genes, I need to understand what the genes are. And I ran into some kind of a lot more complexity and a lot more interesting detail in terms of what's going on under the hood in terms of the way trichomes are organizing the work that they're doing, creating all of these compounds that make the plant so interesting.
So when you say you were originally going to talk about gene networks and now you're talking about gene families, what's the difference?
So gene networks, you're talking about the way genes interact with each other, either in a regulatory system. You've got, genes that are responsible, transcription factors, for example, that are responsible for turning on a particular set of genes, or you've got other genes that regulate what's going on in different ways, either by turning something up or destroying it or whatever. So that's one kind of gene network. The other kind of gene network is a biosynthetic network. Cannabinoids are pretty big molecules, and the way cells handling making a molecule like that is by assembling it from smaller parts, very standard parts that it just makes it part of what we call core metabolism. And so there's a sequence, there's a pathway of genes that's involved in taking these very standard plugged together parts and building very, very specific large molecules like the cannabinoids.
It's almost like an assembly line.
It is a bit like an assembly line.
That's
Only
interesting.
this one, every step is robotic, but there seems to be about 5 different robots that could be working each step, and we don't know that all the robots are the same or have the same priorities. So,
Okay, so that's gene network. So what's a gene family?
so a gene family, you take a particular kind, well, so here's a great example. The cannabinoid synthases are members of a large and very evolutionarily ancient gene family called the berberine bridge enzymes. We call them a family because it's clear that there was way back in the deep past, before the fungi split off from the plants, there was an enzyme, and all of these enzymes, and they've diversified now to do a lot of different things, but they're all related in terms of their history, their lineage back to this original enzyme. So in cannabis, the berberine bridge family contains on the order of about 20 genes that we would recognize as cannabinoid synthases, or very, very likely to be cannabinoid synthases. And then there's a bunch more of these enzymes, some of which are pretty well expressed in trichomes, that we don't know what they do. All we know is that it's probably a molecule that's in the range of the same size as a cannabinoid. So that would be an example of a gene family.
Hmm. It's fascinating. And one of the things I hear so much in talking to Kevin here at Medicinal Genomics and other folks who are kind of in this space is we don't know. How much do we know about the cannabis genome?
Not very much. Every time, I peel back a layer of the onion, it's like, it's a little bit like one of those Russian doll things, except instead of taking it off and there's a smaller doll inside, you take it off and there's 50 smaller dolls, and they're all a little bit different from each other. So everything about this genome is unlike other genomes I've worked with, I slow down a little bit. I'm thinking about the maize genome. Yeah, maybe close to the same level of crazy. But basically, this has been an intensely neglected crop for a very long time. So you compare what we know about the cannabis genome to what we know about, tomato, corn, soybean, for Pete's sake, sweet potato, um, where there's, entire academic careers have been sunk into studying, these other crops. And you come over to cannabis and you really starting with, stone knives and bear claws in terms of the amount that we know. We do have various genomes that are finished to various rates of quality. We do have an initial run at annotating these genomes, which is basically just going through and saying, here's a gene, here's a repeat element, and whatnot. Um, and we're just at the beginning of starting to get a sense for how much the genome varies from plant to plant, from cultivar to cultivar within cannabis. Um, and this is really my focus, getting into specific gene families, like the family of genes that create the early precursors in the pathway to the cannabinoids, or the terpene synthases, for example. How much variation do we see in these from cultivar to cultivar in terms of just whole blocks of these genes being missing? And we're seeing that, new ones that we haven't seen, showing up as we sequence new cultivars, um, and also changes within the genes themselves. I can reliably say, this is the THC synthase, or whatever. But every plant I look at, the gene is just a little bit different. So the amount of variation in this genome, particularly within the parts of the genome that are relevant to the kind of breeding we want to do, um, I, the amount of variation is amazing. That much we know right now. So on the one hand, from the standpoint of trying to study this genome, it's frustrating because I can't just write a simple document that says the cannabis genome is this. Um, um, on the other hand, it's really exciting because every time you turn around, there's something new. And probably most importantly, from a breeding standpoint, breeding is about you're mixing up. You have this line and this line, and this one has these characteristics you like, and this one has these other characteristics you like. You want to create a stable hybrid between them that has all the things that you want. So it's about making combinations of different genetic elements. So if you take a step back and look at this variation rate thing, it means the number of potential combinations, or another way to look at that is the number of possible breeding targets that we can get to with this plant, is incredible. If you compare that to something like soybean, where the amount of genetic variation within, the breeding stock that we have available to us, it's extremely limited. Um, what you can do with that genome, because there's just very little. It's a very, what we call a bottlenecked genetic population. Cannabis is absolutely the opposite extreme. And let's see, to actually answer your question, we are just at the beginning of understanding how much variation happens.
Wow, that's fascinating. And so how much of that genetic variation in cannabis is just because it has been sort of on the fringes and hasn't had as much intense study as, say, a soybean? Or is it something that was always part of the plant, or is it kind of due to circumstance?
So um, that's kind of, really not surprising. It's kind of a complex question. Um, one of the things Um, so if you look at corn, for example, there's an ancestral species called teosinte, and teosinte actually still exists. You can grow this stuff. You wouldn't be able to. There's a way to eat it that has to do with like soaking it in lye and stuff like that. Uh, but there was a set of like a half a dozen mutations that happened in teosinte that turned it into something kind of looked like, modern field corn. Um, and so then we've been breeding on that all these years, and you drive across Iowa, and boy, I'm here to tell you, what we're growing in Iowa right now doesn't look anything like what the Inca and Maya civilizations discovered. Yeah, what is that, on the range of 1,000 years um, ago? So that's an example where we've got the product of a millennium of selection and now targeted breeding, and we've got the wild relative. In cannabis, it seems like all we have is, we don't have really, really good what you would call land races or wild relatives that everybody agrees, yeah, this is what it looked like before human selection. Everything. I mean, we have been doing selection in this species really since the Egyptians. So it's a very, very large number of generations, plant generations have taken place while we've been selecting on this. So we've got kind of this crazy mismatch of, we've been selecting on this genome, and you know we we end up with something that is basically kind of a ways from what the natural genome would look like. The other thing that's going on is there's been a lot of it wasn't random in terms of what individual breeders were doing, but if you kind of like back up and look at the whole effect, it's essentially kind of like random, all of this mixing of Southeast Asian strains, and it's strains found from various places around the world to create new varieties. So we have that aspect of it, as well as that there's basically everything you pick up right now is a hybrid, meaning this is a diploid organism, meaning it's got two copies of the chromosome, and those two copies are very dissimilar to each other. So if I try to like breed this thing, in my F1 population, all of the seeds are going to be different. Um, so that's another aspect where we're very far behind the species, where I mean, corn, I don't know how long, Missouri 17, these old inbreds that have been reliable and understood and studied for 50 years. And we're kind of a ways from that in cannabis.
Right. But I would think the potential to breed a new and interesting strain of corn, that's got to be pretty hard to do, correct?
It is um, to get to something that's really fundamentally different. I mean, what modern breeding in corn is doing, so it's moving to very much more sophisticated mathematical models for looking at at the relationship between observable genetic markers and output traits. Um, but basically, we're on this part of the curve right now where for corn, we're increasing yield by about 1.5% a year.
Wow.
It still looks like the same plant, but 1.5% more than you got last year. And that's been a very reliable trend at about that rate since about 1950 or so. We don't know we're going to hit it. There's got to be a theoretical maximum someplace, but
Wow.
cannabis will be very different from that, because we're just so early in breeding. We don't know, you know there could be we could flail around and fail to find much that's particularly useful I don't think that's going to be the outcome. On the other hand, at the other extreme, we could end up really hitting pay dirt on some new combinations that we haven't, we haven't known to try yet. So maize breeding is in many ways boring and reliable. It's, General Motors is just kind of plugging along. Cannabis breeding is going to be a lot more exciting in terms of what are the kinds of things that we get out of it. I guess the other thing to say there is that in maize, there's two main traits that all of the big breeding companies are focused on right now. One is yield, period, and the second is drought tolerance, or yield under drought. Nitrogen use efficiency or uptake efficiency is another one that's further down on the list. You move over to cannabis, and yield is going to be really important, but it's a very complicated trait to breed with. Drought tolerance is going to be important, but I think that in the short term, the kinds of things we're going to be looking at is creating stable, reliable lines that give you a very specific oil profile under a given growth regime. That's what the market is the most looking for right now, the end user market. And that's also going to be what I think we're going to be best positioned to be able to breed for in the short term.
So you're saying a consistent cannabinoid terpene profile from generation to generation. That's sort of the first hill to tackle.
Yeah, that's the first one that we're going to be able to go after. Now, if you talk to growers, I mean, there's a lot of growers out there who say, man, I got this one variety and I can sell every gram of this stuff that I can grow, and I can sell it at a big premium, but I can't afford to grow it because the overall yield is so low. What can be done about this? And there will be things to do there, but anytime so overall yield, we don't even know how many genes are involved in that. And so it's much more complicated to design a breeding program that, and it may be it's probably going to end up modeled very closely on what's currently been done in maize. And it'll just be gradual incremental increases in yield year by year.
Yeah, I mean, and that's fascinating when you said that, corn yield is increasing by 1% a year and has been since 1950. So
yeah,
I can only imagine, 70 years from now what cannabis is going to look like.
Yeah.
It's fascinating. It's fascinating. So in terms of cannabinoids and terpenes, I got to imagine that's intensely complicated as well, just because, I mean, modern testing labs can't even test for all the terpenes that the plant is creating.
Yeah.
So where do you start there?
So the terpenes are very difficult for a number of reasons, as a breeding target, as a, any way you want to go at it. One of the big challenges with the terpenes is they're volatile. I mean, they just evaporate. And if you take your, you got a whole bunch of fresh bud, and your curing protocol is to put it in basically a low temperature oven for a day or two and just drive all the water out, it's going to be nice and dry, and you're going to lose almost all of your terpenes out of there. so there's all these variables before your sample even gets to the extraction step in your testing lab, that impact, the terpenes, they all boil at different points. The monoterpenes are boiling in the like 150 degrees Celsius range, and the sesquiterpenes are larger, and you're more like 225 or so. But but what that means is different molecules are being depleted out of your sample at different rates. So, if you're looking at the ratio of myrcene to caryophyllene, for example, it's going to change depending on how you cure the plant and how old the sample is. So it's kind of a nightmare scenario for a breeding program where you would need to be going in and periodically checking, do I have exactly the terpene, profile that I want? It's just, it's, it's inherently difficult just because, uh, this kind of experimental variability is built in. The other aspect of this is that this is a particular area of chromatography where you've got very complicated mixes. People are dealing with different matrices that they have to do the extraction from, um, and there's a lot of stuff that makes it difficult to do these measurements really well. Um, Steep Hill seemed to do a particularly good job of it, but your mileage is going to vary because this stuff is not easy, and we haven't standardized to like a global set of best practices that everybody would adhere to.
Yeah, absolutely. When I speak with folks in the lab testing side, which is one of the focus areas of CannMed, that's one of the things they come back to all the time is the need
Yeah,
for a standard to sort
yeah,
of get consistent results from lab to lab. So hopefully we'll see that soon.
yeah.
One of the things you sort of touched on, and it's not an exact correlation, but I did want to bring up to you this whole idea of nature versus nurture as well,
Hmm.
where the genes are only going to do so much. A lot's going to depend on the environment you place it in, the type of lights that you're providing. So I was wondering if you could comment a bit on that, and is there variation even between strains where some are more sensitive to sort of those changes, or is it universal?
A little bit of all of those.
Of course.
Yeah. So for a given genetic background, you have you grow up this stuff, and either it's completely genetically pure. Well, we don't have that, so you do cuttings. Um, we already know, so you grow them indoors and outdoors. You grow them under high pressure sodium, or with an LED light. You grow them outdoors in 5 different fields, or you walk a field outdoors and just look at if you've got a lot of variation in the field. And most of the time when I'm out in a field, it's rare to find a field that is really 100% homogeneous in terms of soil type, the amount of water that the plants have available to them, and the various things that, shading from wind, for example. Um, so a given genetic background is going to give you basically like a set of performance characteristics. It does this under these lights and this under these other lights, and this range of things when grown outdoors. The one thing for sure is it's going to be really different outdoors than it is in the greenhouse. But to come back to that last part of your question, that's something that's going to vary from strain to strain. Some strains, I don't know. Some strains are going to be really good indoors, and they're just going to be crap outdoors, or the opposite. You're going to have other strains that are really a lot less sensitive to environmental changes. Um, than others. And it all comes down basically to a complex interplay of the set of gene networks working in each particular plant. One of the, if from a data analysis perspective, what you would most like to have, rather than say, here's the plant and it performs like this, what you would have is a complete breakdown. Here it is in 5 different growth environments and a half a dozen replications of each one. So within each growth environment, how much does it vary and how much does it vary between growth environments. The cost of doing something like that starts to get fabulous very, very quickly. So you have to work with less data than that.
Yeah. And now, is that something that could eventually be sort of bred into the plant, is to kind of have, I guess, more robustness so they're not so sensitive to those variations, like you mentioned out in the field?
Yeah, but targeted breeding like that, you need to know basically what are the exact genes, or at least what are the chunks of the genome. Sometimes you can do a mapping and it just says, well, it's this part of chromosome 8 is what's giving us this behavior. But you need to know something like that. And then once you have that, you can devise a molecular marker and you can follow it through your crosses and breed for, in this case, robustness to environmental change. So there's a prior step first.
Gotcha. One of the things you mentioned was, that a lot of commercial cultivation right now is done with cuttings. And I imagine the goal is to eventually get to stabilizing seeds for propagation. So how far away do you think we are from that?
So there are some lines on the hemp side that are relatively homogeneous. Homozygous is the exact term. But if we do it the traditional by the traditional method, which is years of what's called back crossing, targeted back crossing, where you're monitoring, am I fixing, meaning making this homogeneous in the genome? Am I fixing all the positive gene variants, the alleles? And not, what you want to avoid is fixing negative alleles, which is, because what you're creating is an inbred line. And the danger with inbreds is you fix deleterious genes in there. So the magic of making good inbreds is being able to follow, knowing enough about the genome that you can breed something that's inbred and doesn't have serious deleterious alleles in there. So the old-fashioned way to do that takes years. Depending on who you talk to, you're going to get different numbers on how long it would take. But if you were just to do, say, a half dozen back crosses, you're going to be a fair amount of the way there. You could do that probably in 2 or 3 years. But to get to really good homogeneity is going to take longer than that. Now, on the other hand, there is a trick called double haploids, where you start with a haploid cell, like, for example, a pollen grain, and you trick it. It's a chemical trick that you can do and regenerate a diploid plant from it. That is, now what you've done is you forced it to go through a doubling without creating 2 cells, and bang, now you've suddenly got a line that is exactly homozygous at every position. that's called double haploid technology, and it doesn't exist yet in cannabis. Um, we don't technically know if it's possible in cannabis yet. At Syngenta, we got it going in maize, and it was um, a nuisance, but we had it running. Then we tried, and we had very good experts working on it. We tried to get it going in tomato, and just failed abjectly. We ended up licensing from another company in the Netherlands that had gotten it working. So cannabis may be, it's going to be really hard to get it working. It could be really easy, or it could be impossible. But with double haploids, that would revolutionize a lot of things in the industry very quickly.
So you're actually taking pollen and having it essentially breed with itself?
Basically.
It's fascinating. So I would be remiss if I didn't ask you about your position at Front Range Biosciences. What's your role there? What are the things that you're working on?
So I am the director of bioinformatics. We're a little startup, so that's a fancy term for basically the doing the main amount of the bioinformatics work, which is mainly focused on gene discovery. Developing the catalog of the genes that we'll care about for our breeding purposes.
And so what is your focus currently?
So I've been focused on the terpene family, and right now I'm working on the cannabinoid family, both the what we call the terminal synthases that make the individual cannabinoids, and the upstream parts of the pathway. There's a number of other things that along the way we're going to get to. Disease resistance obviously is one that there's a critical need for in cannabis, but you know basically, I mean, right now I'm really focused on the cannabinoid pathway.
Excellent. And any cannabinoids in particular, or is it sort of a combination of the tried and true THC, CBD, or are there sort of other lesser cannabinoids that you're interested in?
Yeah, we're mostly interested in the lesser cannabinoids. In the so Front Range is focused on CBD dominant, legally hemp varieties, and so one of the things that everybody in the field is very concerned with is not having your plants go hot. That 0.3% THC.
Of course.
So understanding what are the genes that are involved in creating this extraneous THC. A lot of times you're seeing this in varieties where the THC synthase gene looks like it should be stone dead. So there are other sources of THC. So understanding those, but then also there's a lot of interest in the other cannabinoids, and for very good reason. I mean, if you only know one thing about pharmaceutical chemistry, it's that if you've got a family of related molecules and you know that 2 or 3 of them are really super interesting, you better go look at the rest of them. So cannabichromene is one of the more easy to find, but also CBG um, um, strains, the varin, cannabivarin, and THCV, and there's a number of these, lesser cannabinoids that we're looking for plants that make reasonable amounts of them, but there's a lot of perfectly good looking synthase genes that are actually expressed in flowers, not at the same rate as the CBD and THC synthases, but these are real healthy genes, and we'd like to know what do these things do.
Excellent. So you're very much focused on creating, high yield resinous buds for terpene and cannabinoid production. Any interest in sort of looking into hemp as a textile or for other uses beyond sort of the pharmaceutical or nutraceutical aspects?
Yeah, definitely. There's so the there's various sides to, like, the economic argument in terms of, what is it profitable for the farmer versus other things that they could be doing, um, and so that kind of drives, like, where are the most important market segments for us to be looking at. But hemp as a fiber, um, it's got this long list of really substantial advantages, and if nothing else, creating varieties where you get, say, both things going on at the same time. You've got,
That'd be nice.
you have very good oil production and very good fiber production. That's something that we've talked about. I think it's maybe more likely that it's going to continue to be two very different kinds of varieties. But the short answer to the question is, yeah, that's that's obviously another one. That's the other is use of the seeds as a protein source.
Absolutely.
So.
Well, that's very fascinating stuff, Keith. Where can people learn more? Where can people learn more about your work and about Front Range Biosciences?
Well, let's see. You can go to the Front Range Biosciences website. For my terpene work, I've got a paper out in PLOS One. You can go to CannMed. I understand I'll be giving a talk.
Yes, please.
Yeah, and I'm starting to kind of shape out the profiles of another paper that will be focused on the cannabinoid pathway.
Excellent. When can we expect to see that?
Oh, I'm not going to make any kind of guess on that, but within the year, I should.
All right, excellent. Well, we'll be sure to share that with everyone, and hey, hopefully it'll be there in time for CannMed. We can get a glimpse into it. So, thanks again, Keith, for taking the time to talk to us today.
No problem.
We'll see you in Pasadena this fall.
See you later.
All right.
I hope you enjoyed my conversation with Keith Allen. Check out the links in the show description to learn more about the topics we discussed, and please also check out the Front Range Biosciences website. Our next episode will drop August 5th. In the meantime, please go to cannmedevents.com/coffeetalk and sign up for email updates. That will enter you into a drawing to win 2 tickets to our CannMed 2021 VIP dinner, and also keep you up to date with all things CannMed 2021. If social media is your thing, you can find us on Facebook, Instagram, Twitter, and LinkedIn. Just search for CannMed Events. And lastly, if you are listening via a podcast app, go ahead and hit the subscribe button so that new episodes automatically download to your device. And please also leave us a 5-star review. All right, that's it from us. Stay safe, stay healthy, and be sure to come back for the next episode of CannMed Coffee Talk.